Hello, my name is Garth Humphreys, Principal Ecologist at Biota Environmental Sciences, here today to talk about the stygofauna baseline report undertaken for the Strategic Regional Environmental and Baseline Assessment of the Beetaloo Subbasin. The Northern Territory Government is undertaking the Strategic Regional Environmental and Baseline Assessment, or SREBA, of the Beetaloo Subbasin. It's the area shown in red at left there. This study we're talking about today focused on stygofauna as part of the SREBA research program looking at aquatic ecosystems. The work was undertaken by Biota Environmental Sciences and Helix Molecular Solutions, both specialists in subterranean fauna. Stygofauna are subterranean aquatic animals that live in groundwater systems. In evolutionary terms, they've been in these habitats for a very long time and have become completely adapted to life there. They have features such as depigmentation, so complete loss of pigment or very pale pigment, eyes reduced or absent, and quite elongate body morphology, as you can see from those example photos. Work completed elsewhere in Australia has shown that Northern Australia has a diverse stygofauna in suitable habitats, and also that some species can be restricted in distribution to particular hydrogeological units. So our first step with undertaking the SREBA stygofauna study was to establish what information already existed and what work had previously been completed. So that desktop review collated all the available data on stygofauna in the SREBA study area, and it revealed that there's been very little past sampling done. Mostly historical work has been limited to ad hoc collecting in caves or some groundwater bore sampling, with only one substantive study undertaken by CSIRO in collaboration with Charles Darwin University and La Trobe University. The records that we could collate showed a fauna typical of what we see in Northern Australia, which is dominated by crustacean orders, and in particular the decapoda, which includes prawns and crabs and 10-limbed crustaceans. The other component to the desktop review was to examine what subterranean habitats might be present in the SREBA study area. And the most important aquifer was the Cambrian limestone aquifer, which is an extensive regional groundwater system. It has several hydrogeological formations within it, particularly the Tyndall, Gumridge, Montigny and Anthony Lagoon units. Based on structural characteristics of the rocks, the Tyndall limestone a priori was the most likely to represent stygofauna habitat. Previous work has shown that it's a highly transmissive habitat, it's widely connected in terms of groundwater flow, has space for fauna below the water table, so actual habitat space for animals to live, and it has karst features such as solution tubes and caves that provide rapid recharge during flood events from surface sources. So our next step then was to undertake the sampling design for the survey, and that was based on selecting from existing groundwater bores. And these have all been put in for other purposes, be that water exploration for water supply or for pastoral uses or other reasons. And in doing the selection, we followed a set of criteria, which was to attempt to obtain adequate spatial coverage across the SREBA extent. And we stratified our sites amongst the hydrogeological formations we knew to be present. And we aim to select a number of sites that would overall characterise the stygal assemblage of the study area, so we would get a reasonable idea of the species present. And that's been informed from what's been done elsewhere in Northern Australia in regards to the number of sites and effort. And 2 phases of sampling were collected, one post-wet season. As we know, groundwater recharge events can alter the results and sometimes result in different species being collected. So following those approaches, we ended up with 91 sites. 66 of those could actually be sampled. Some of the sites, unfortunately, are blocked or land access proves difficult. They cannot be located at the coordinates. So we always have a series of limitations with this type of work. It's not specific to the SREBA study. And of the 66 sites that were sampled, most were in the Daly Formation and most of those were in the Tyndall Limestone unit. And that reflects simply just where the number of bores were maximised within the study area. And whilst it's a bias in the effort, it actually biases it towards the habitat type that we expected would provide the most number of animals. In terms of the actual methods and techniques that we use, Sampling's carried out with custom-built plankton nets. These are lowered down groundwater bores and hauled up through the water column a minimum of 5 times at each site, so we consolidate that into a single sample from that site. It's a little bit like fishing, effectively. The samples are then sorted under dissecting microscopes by zoologists experienced in this type of work, and we recover all the stygofauna at that stage and do some preliminary identifications based on morphotype to place them into orders and families. The specimens were preserved in 100% ethanol, which is useful for morphology, but also importantly maximises their use in DNA analysis, molecular analysis. This is an area where there's been little past work in the Northern Territory, so we knew that would be important to place these animals into context and work out how many species were present. Habitat data was also collected, so each bore was logged with a groundwater probe that collected data on physicochemistry, so temperature, salinity, other parameters like that. And we also collected groundwater samples from each site and processed and preserved those to provide for environmental DNA analysis, or eDNA analysis. That's a lab assay that picks up residual DNA shed into the environment from animals, so it's a way of indirectly detecting what species might be there, even if you don't catch them while you're doing the fishing. So what did we find in terms of results? Stygofauna were recorded from 23 of the 66 sites, so about 35%. And the assemblage that we picked up overall comprised at least 28 species level taxa, or 28 species effectively. As we expected, the fauna was strongly dominated by the crustacea. 7 of the 8 orders that were represented amongst the animals were crustaceans. And in particular, the copepods and the decapods accounted for over half the specimens collected alone. The DNA analysis was carried out on all the animals that we collected, and it was very clear that almost all of these 28 species are newly recorded species, and they were very divergent from all the existing reference data that's available. So there's no likelihood that any of these animals actually represented species that are known elsewhere in Northern Australia. One group in particular that we focused on was the decapoda. In this study found to be the most widespread stygofauna group, and in particular the decapod Parisia unguis. That had also been found to be widespread by the previous work conducted by CSIRO in collaboration with the universities. Our DNA analysis of the decapod specimens found 6 distinct lineages amongst the specimens. They were divergent, but not quite to the level that we would expect for species, but clearly they have been subject to some degree of genetic isolation. 5 of those were assigned to the nominal species Parisia unguis, which is known to be quite widespread in the locality. And the last was actually belonging to a different genus based on the phylogenetic analysis, Caridina, and it did not correspond to any of the known Caridinas, so nominally Caridinus beetaloo. And as you can see from the table, there was some evidence of potential geographic structure as well. So some of the Parisias were widespread. Group 5 at the bottom there was recorded from 3 of the 4 units. And at the moment, some of them, like Parisia split 1 and 2, those are only limited to the Daly Basin and the Tyndall Limestone Formation in particular. Noting, of course, that with stygofaunal work, there are often strong ecological sampling effects, and additional sampling would be required to really prove up this model. And it may also help us to untangle some of the potential taxonomic difficulties with this particular group and get a better idea of whether these are actually valid species that we're seeing in these 5 different groups, and whether there's any evidence of what is causing the separation genetically. So in regard to the environmental DNA work, or eDNA work, we were specifically interested in Parisia unguis, and during the course of this study we developed and validated an eDNA probe, a specific assay that responds to that particular taxon, and that was used on all the filter papers that we collected from all the sites that had the groundwater pumped through them. And what we found with that work was that 32 of the 66 sites amplified for the presence of the Parisia split DNA, so we had a positive hit for environmental DNA. At the left there, they're shown with the blue circles with the dot in the middle. And at several sites, we also had collected Parisia specimens as well. So we had high confidence that that actually corresponded to a valid result. And there were a number of lab protocols followed as well to validate the sensitivity of the probe and to look at how long water samples could yield DNA from that particular taxon after animals were removed. So we had high confidence that the qPCR approach that we developed for the eDNA work was working well. And we collected eDNA data for Parisia from 25 additional sites beyond those where we actually collected animals from conventional haul net sampling. So that really significantly expanded the mapped distribution of Parisia unguis nominally. And it demonstrates that that Parisia taxon occurs across all formations of the study area and really highlights the value of this environmental DNA work as an adjunct survey method. One element that we like to look at with subterranean fauna work is to look at diversity within habitats and in particular within particular hydrological formations. And what we found was consistent with our expectations that the highest diversity was in the Tyndall limestone. 26 of the 28 species that we got overall in the study actually occurred in that particular habitat. The Tyndall formation also contained the top 8 most species-rich individual sites, including one borehole at the left there, which had 10 species from the single site, another with 8 and another with 6. So these are quite diverse results from a single site for just 2 phases of sampling. And the high proportion of fauna and the diversity of fauna in this unit probably reflects things like shallow depth to groundwater in the Tyndall limestone, and that structurally it's very suitable in terms of having a lot of cavities and spaces and solution tubes that are flooded below the water table, so there's lots of space for the animals to actually occur, and there's a high degree of organic input during recharge events from the surface. Habitat conditions in the groundwater for stygofauna. On average in the study area, the groundwater systems are warm, low 30 degrees, slightly brackish, so a little bit above what would normally be considered fresh water in terms of salinity. Neutral, almost exactly on pH 7, and relatively low in dissolved oxygen. And these sort of broad parameters are consistent with what's been seen elsewhere in groundwater habitats in Northern Australia. What was interesting is that our data showed significant changes post the wet season. So comparing our phase 1 in the dry to phase 2, which was after the wet, the salinity dropped to less than half of that, presumably due to the influx of fresh water from surface sources. And the pH also became more basic, as well as some other minor habitat changes as well. So these recharge events are clearly important in biophysical processes that affect stygal habitat, and it's one of the key drivers for bringing organic material and surface-derived energy into these subterranean systems to drive the bottom level of their food webs. In terms of overall conclusions, the stygofauna assemblage of the Beetaloo subbasin has now been significantly better documented. Previously there were 11 species known in the area. Now there's at least 38 species as a result of this work. That's treating the Parisia conservatively as a single species still, even though we know there's a number of genetic types within it. Our analyses of the sample data to look at the potential total species present, so the data can be used to predict how many species there are in total, suggest that perhaps there could be double this number, so up to 50 or 60 species at least. And that's again consistent with what's been seen in other parts of Northern Australia at this scale, where you have large limestone aquifers that are suitable habitats. The findings of the work really highlight how important the Cambrian limestone aquifer is as stygofauna habitat, and the Tyndall limestone unit in particular. So the maintenance of this aquifer habitat and the processes that sustain it, such as groundwater flows and recharge events from surface sources, is critical to ensuring the persistence of this diverse stygofauna community that's present in the Tyndall limestone.